Welcome everyone. I'm excited to kick off today's presentation on oxidative phosphorylation. This is the first slide, so I want to give you a quick overview. We'll be exploring how cells produce energy through this fascinating process. We'll cover the key mechanisms, the role of mitochondria, and why this process is vital for life. So, let's dive in and discover the details together. Let's take a closer look at the respiratory chain complexes I, and IV. These complexes are responsible for coupling electron transfer with H+ ejection from the mitochondrial matrix into the intermembrane space. This process is crucial because it helps establish a proton gradient that the cell can later use to produce energy. Now, because the outer mitochondrial membrane contains large channels, these protons can sometimes equilibrate with the cytosol. This means that while protons are being pumped into the intermembrane space, some may diffuse back out, especially through these channels. On the right, you can see the conventional view of mitochondrial structure. The respiratory chain is localized specifically in the cristae of the inner membrane. These folds increase the surface area, providing more space for these complexes to work efficiently. The key point here is that the spontaneous transfer of electrons through these complexes is linked to H+ pumping. This process conserves part of the free energy from the electron transfers as potential energy, specifically as an electrochemical H+ gradient. This gradient is essential because it is used by ATP synthase to generate ATP, powering many cellular activities. So, in essence, the respiratory chain is not just about moving electrons. It's about creating a stored form of energy that our cells can harness when needed. Let's take a closer look at these 3D reconstructions based on electron micrographs of isolated mitochondria. These images were captured with a large depth of field, which means we can see the structures in greater detail across different angles. By tilting the mitochondria at various angles, we've been able to visualize the complex architecture of their inner membrane. What's particularly interesting is that the infoldings of the inner membrane are quite variable in shape, indicating a dynamic and adaptable structure. These infoldings are not isolated. They are connected to the outer regions of the mitochondria and to each other via narrow tubular regions. This connectivity is crucial because it suggests a highly interconnected network within the mitochondria, facilitating efficient communication and material transfer. Overall, these findings highlight the intricate and flexible architecture of mitochondria, emphasizing how their structure supports their vital functions in energy production and metabolic regulation. Here we see an electron micrograph of a neurospora mitochondrion, captured by Dr. C. Manella. It's taken from a frozen sample, and importantly, no fixatives or stains were used, so we're seeing the mitochondrion as it naturally appears, without any artifacts that might distort its internal structures. This helps us really understand its true architecture. Now, one key feature to note is the tubular cristae. These structures are connected to the inner mitochondrial membrane through narrow passageways. These passages are significant because they can actually limit how quickly protons, H+ ions, can move between the lumen of the cristae and the intermembrane space. This has implications for how efficiently the mitochondria can generate and maintain the proton gradient needed for ATP production. Additionally, there's evidence suggesting that when protons are pumped out of the mitochondrial matrix, they don't immediately disperse into the bulk phase of the intermembrane space. Instead, they tend to spread along the surface of the membrane, which carries anionic properties. This surface spreading means that protons equilibrate more slowly, effectively maintaining a higher H+ gradient across the membrane. Connecting this to our overall theme, these structural and functional details highlight how mitochondria are finely tuned to optimize energy production. By controlling proton movement and creating a sustained gradient, they can maximize ATP synthesis efficiency. This interplay between structure and function is a fundamental aspect of mitochondrial bioenergetics. So let's talk about how electrons move spontaneously through the complexes in the respiratory chain. When electrons pass through complexes I, and IV, This process is coupled to the ejection of protons, or H+ ions, from the mitochondrial matrix. This coupling is really crucial because it creates a proton gradient across the inner mitochondrial membrane, which is essential for ATP production. Now, for every 2 electrons transferred from NADH to oxygen via this chain, about 10 H+ ions are ejected from the matrix. That's a significant number, and it highlights how efficient this process is at generating a proton motive force. We'll also look at the specific H+E ratios for each complex. These ratios tell us how many protons are moved for each electron transferred. Understanding these ratios helps us grasp how each step contributes to the overall process of energy production. Connecting this to the bigger picture, all these steps work together seamlessly to convert the energy stored in nutrients into a form our cells can use, ATP. It's a finely tuned system, and these proton movements are at the heart of that energy conversion. Let's take a closer look at complex I, also known as NADH dehydrogenase. This enzyme plays a crucial role in the electron transport chain. When NADH donates 2 electrons to complex I, it facilitates the transfer of these electrons to CoQ, or ubiquinone. An important point here is that for every 2 electrons transferred, complex I pumps out 4 protons, or H+ ions, from the mitochondrial matrix into the intermembrane space. This proton pumping is vital because it helps establish the electrochemical gradient that ultimately drives ATP synthesis. So, in essence, complex I not only transfers electrons, but also contributes significantly to the proton motive force, which is the energy source for ATP production in the mitochondria. Understanding this process helps us see how energy from nutrients is converted into usable cellular energy. Let's talk about one of the main challenges in understanding how complex I functions. The lack of high-resolution structural information for its membrane domain has really hindered our ability to fully elucidate how H+ transport actually occurs. Without detailed images, it's difficult to pinpoint exactly how protons are moved across the membrane. Now, an important point to consider is that direct coupling between transmembrane H+ flux and electron transfer is unlikely. This is because the electron-transferring prosthetic groups, like FMN and the Phes clusters, are located in the peripheral domain of complex I, away from the membrane itself. So, it's not a straightforward one-to-one process where electrons and protons are directly linked at the same site. This leads us to an interesting hypothesis, that protein conformational changes are involved in H+ transport, similar to how ion pumps work. Instead of a direct transfer, the enzyme might undergo structural shifts that facilitate proton movement across the membrane. This concept aligns with the overall theme of how complex I converts electron energy into a proton gradient, but highlights that the mechanism is still not fully understood, largely because of the structural data limitations. Now let's turn our attention to complex III, also known as the BC1 complex. An important aspect of this complex is how it handles proton transport, and here, Coenzyme Q, or CoQ, plays a central role. CoQ acts as an electron carrier within the inner mitochondrial membrane, shuttling electrons from complex I and II to complex III. But beyond just moving electrons, CoQ is also involved in the process of proton translocation across the membrane. This movement of protons is crucial because it helps establish the electrochemical gradient that drives ATP synthesis. So, understanding how CoQ functions in this context is key to grasping the overall efficiency of cellular respiration. It's a perfect example of how interconnected electron transfer and proton pumping are in energy production. The Q cycle relies on the mobility of coenzyme Q, or ubiquinone, within the lipid bilayer of the mitochondrial membrane. This movement is crucial because it allows Q to shuttle electrons efficiently between different complexes in the electron transport chain. There is also strong evidence that these electron transfers happen one at a time, meaning one electron moves at a time rather than all at once. During this process, an intermediate called a semiquinone radical is formed, which is essentially a partially reduced form of Q. Understanding this stepwise transfer and the role of the semiquinone is key to grasping how the electron transport chain works so efficiently. It highlights the importance of Q's mobility and the controlled nature of electron transfers, which ultimately drive ATP synthesis, connecting this to the overall theme of how cells generate energy through complex, yet highly coordinated processes. Today, I want to walk you through one version of the Q cycle, which is a crucial part of how electrons are transferred within complex III of the electron transport chain. So, electrons enter complex III via a molecule called coenzyme QH2, also known as ubiquinol. Now, this coenzyme QH2 binds at a specific site on the positive side of the inner mitochondrial membrane, right next to the intermembrane space. This positioning is important because it allows the electrons to be efficiently transferred into the complex, facilitating the process of oxidative phosphorylation. The Q cycle essentially helps us understand how electrons move smoothly through the chain, ultimately leading to the production of ATP. So, by focusing on where coenzyme QH2 binds, on the positive side of the membrane, we can appreciate how the structure of the mitochondria is perfectly arranged to maximize energy transfer. This connection between the binding site and the overall function of the electron transport chain highlights the elegance of cellular respiration. Now, let's look at what happens when QH2 donates electrons. It gives up one electron to the Risk iron-sulfur center, often called Phes. This transfer is crucial because it initiates the next steps in the electron transport chain. The Phes cluster then gets reoxidized by passing that electron to cytochrome C1, which in turn passes it out of complex III to cytochrome C. An interesting point here is that losing just one electron from QH2 creates a semiquinone radical, represented as Q. It's a sort of intermediate state. Initially, this semiquinone might still hold onto a proton, forming QH, which can influence how electrons and protons are transferred during respiration. Understanding this process helps us see how electrons flow efficiently through the chain, ultimately driving ATP synthesis. It's a fine example of how coordinated electron transfer and proton movement generate the energy our cells need to function. Now, let's look at what happens with the second electron. It's transferred from the semiquinone to CytBl, which is heme Bl from there. It moves through CytBh, crossing the membrane to reach another CoQ molecule that's bound at a site on the matrix side. This process is crucial because it helps us understand how electrons are efficiently passed along in the electron transport chain. As this second electron is transferred, the fully oxidized CoQ, which was generated when the second electron was passed to the B cytochromes, can then dissociate from its binding site. This site is located near the intermembrane space, and the dissociation is important because it allows CoQ to participate in further redox reactions elsewhere. It's also key to note that during this two-electron oxidation of QH2, two protons are released into the intermembrane space. This proton release is a vital part of creating the electrochemical gradient that drives ATP synthesis. So, in summary, this slide highlights the movement of electrons through the B cytochromes and the associated proton transfer, which together contribute to the overall process of energy conversion in mitochondria. This step exemplifies the intricate coordination between electron transfer and proton movement, underpinning the entire chain's efficiency. In this slide, I want to introduce an alternative mechanism that has been proposed to explain how electron transfer occurs within this system. Instead of the traditional step-by-step process, this model suggests that the two electrons are transferred simultaneously. Specifically, from QH2 to both the Phes cluster and cytochrome Bl at nearly the same time. This concept is quite interesting because, by doing so, it could eliminate the formation of the semiquinone intermediate, which is usually observed in the stepwise transfer process. The key idea here is that this simultaneous transfer might streamline the process, making it more efficient. Understanding this alternative mechanism is important because it helps us explore different ways that electron flow can be managed within the electron transport chain, which ties back to the overall theme of how these systems optimize energy conversion. It challenges our traditional view and opens the door to new insights about the dynamics of electron transfer. So it takes about 2 cycles for CoQ, which is bound near the matrix, to be reduced to QH2. This process happens as CoQ accepts 2 electrons from the B hemes, and at the same time, 2 protons are extracted from the matrix compartment. Essentially, in just 2 cycles, 2 molecules of QH2 enter the pathway, but interestingly, one of them gets regenerated in the process. This is a crucial step because it highlights how electrons and protons are transferred efficiently within the electron transport chain, ultimately driving the production of ATP. It's a fine example of how the system maintains a balance, recycling molecules while moving electrons forward. Let's take a closer look at the overall reaction catalyzed by complex III, which is also known as the cytochrome BC1 complex. The key point here is the Q cycle, which is essential for electron transfer and proton pumping. So on this slide, we see that the reaction involves QH2, or ubiquinol, donating electrons. When QH2 is oxidized, it releases 2 protons into the intermembrane space. That's what we see as the 4H+ outside. Simultaneously, electrons are transferred through the complex to cytochrome C, reducing it from Fe3+ to Fe2+. The net effect is that for every 2 electrons transferred, the complex releases 4 protons into the intermembrane space, contributing to the proton gradient that drives ATP synthesis. This overall process is crucial because it links electron transport to proton translocation, which, as you remember, is fundamental to the cell's energy production. The Q cycle efficiently couples these steps, making complex III a vital component of the mitochondrial respiratory chain. Now, let's look at what's happening with protons in this part of the electron transport chain. While 4H+ ions appear outside per net 2 electrons transferred in 2 cycles, only 2 H+ ions are actually taken up on the matrix side. This means there's a sort of imbalance or uncompensated proton movement. In complex IV, we see a similar situation. Here, there's also an uncompensated proton uptake from the matrix side, specifically 4 H+ ions per O2 molecule, or 2 H+ ions for every 2 electrons transferred. The key idea is that these proton transfers aren't always perfectly balanced on both sides of the membrane, which has important implications for how we understand the proton motive force and energy conservation in these processes. It highlights that the system is inherently asymmetric, and this asymmetry is crucial for driving ATP synthesis. So, this uncompensated proton movement is a fundamental aspect of how these complexes contribute to the overall energy conversion in cellular respiration. So what we're seeing here is that for every 2 electrons that pass through the chain, there are effectively 2 protons, or H+ ions, transported across the membrane. This process involves a combination of complexes III and IV working together. What's really important to highlight is that these complexes don't work in isolation. They coordinate to move these protons efficiently. In the diagrams, especially those illustrating H+ to E stoichiometry, you'll notice how complex III plays a key role in this proton translocation. Understanding this coordinated transfer is crucial because it ties directly into how the electron transport chain establishes a proton gradient, which ultimately drives ATP synthesis. So, this step, involving complexes III and IV, is fundamental to the overall energy production process in the cell. Now, here we see half of the homodimeric structure of complex III. You can get a sense of its overall shape and organization. The approximate location of the membrane bilayer is also indicated here, which is crucial because it shows how this complex is embedded within the mitochondrial membrane. What's really important to note, though, is that we're not showing the CoQ binding sites near heme BH and heme BL on this slide. These sites are essential because they facilitate the transfer of electrons. Speaking of electrons, the B hemes are strategically positioned to create a pathway for electrons to move efficiently across the membrane. This arrangement is vital for the proper functioning of the electron transport chain and ultimately for ATP production. So, in the context of the overall theme, understanding the structure and electron pathways in complex III helps us see how energy conversion is tightly organized at the molecular level. Let's take a closer look at the domain with the attached RISC-FES cluster. You can see that it has a flexible link to the rest of the complex, which is really important for its function. The FES protein itself is highlighted in green here. Now, during electron transfer, the position of the FES cluster actually changes. After it extracts an electron from QH2, it moves closer to heme C1, allowing the transfer of that electron. This movement is crucial for efficient electron flow within the complex. I encourage you to view the animation to see this dynamic process in action. It really helps to visualize how these conformational changes facilitate electron transfer in the electron transport chain. After the first electron transfer from QH2 to the FES cluster, there's an interesting event that happens. The CoQ semiquinone is thought to actually shift its position within the Q-binding site. This movement brings it closer to its electron acceptor, which is heme Bl now. Why is this important? Well, this positional change helps to prevent the transfer of a second electron from the semiquinone to the FES cluster. Essentially, this shift acts as a regulatory mechanism, ensuring that electron flow proceeds in a controlled manner and avoids unwanted side reactions. It's a clever way the system maintains efficiency and prevents overreduction, which is crucial for the overall function of the electron transport chain. Complex III is an obligate homodimer, meaning it always functions as a pair of identical subunits working together. One of the key features here is that the FES cluster in one half of the dimer can interact with molecules like CoQ and heme C1 that are bound in the other half. This crosstalk between the two halves is crucial for the electron transfer process. You'll notice the arrows pointing at the FES cluster, which is shown in white or gray, and the heme C1, highlighted in blue or green, in the other half of the complex. This visual helps us understand that even though the two parts are separate, they are interconnected and coordinate their activity to facilitate efficient electron flow. Connecting this to our overall theme, it underscores how structural organization directly impacts mitochondrial function and energy production. Now let's take a look at complex IV, also known as cytochrome oxidase. Here, electrons are donated one at a time by cytochrome C, which binds from the intermembrane space. As these electrons move through the complex, they pass via copper and heme groups, specifically Cu-A and heme, toward the binuclear center. This center is buried deep within the complex and is where the critical reduction of oxygen occurs. The reaction involves 4 electrons, 4 protons, and 1 oxygen molecule, resulting in the formation of 2 water molecules. It's important to note that the protons used in this process are taken up from the mitochondrial matrix. This entire process is a crucial step in the electron transport chain, ultimately driving the production of ATP by facilitating efficient energy transfer. Let's take a closer look at how complex IV contributes to proton pumping. In addition to the protons that are used in the reduction of oxygen to water, there's also a process linked to electron transfer that actively moves protons across the membrane. Specifically, for every 2 electrons transferred, 2 protons are transported from the mitochondrial matrix to the intermembrane space. Over the course of 4 electrons, this amounts to 4 protons being pumped. This proton movement is crucial because it helps establish the electrochemical gradient that drives ATP synthesis. So, overall, complex IV not only facilitates the reduction of oxygen, but also plays a key role in maintaining the proton gradient necessary for energy production. This highlights how interconnected these processes are within cellular respiration and energy generation. Let's talk about how protons move through complex IV. Structural and mutational studies show that protons don't just diffuse randomly. Instead, they pass through specific pathways called proton wires. These wires are essentially chains of groups that can accept or release protons, allowing efficient transfer. Most of these chains are made up of buried water molecules, which form a sort of relay system, along with amino acid side chains and the propionate side chains of hemes. What's fascinating is that there are separate H+ conducting pathways that connect each side of the membrane directly to the buried binuclear center, where oxygen reduction actually happens. These pathways are known as the D and K channels, named after key amino acids, aspartate and lysine residues, extending from the mitochondrial matrix all the way near the binuclear center deep inside complex IV. Understanding these pathways is crucial because they illustrate the precise molecular mechanisms that enable proton transfer and energy conversion in cellular respiration. For visual references, you can look at images from IBI and Crofts that depict these proton wires and pathways. So, what we're looking at here is a switch mechanism that is controlled by the reaction cycle itself. This switch essentially helps transfer a proton from one half wire or half channel to the other, which is crucial for the process. Now, it's important to understand that there can't be an open pathway for H+ completely through the membrane, because if that happened, oxidative phosphorylation would be uncoupled. In other words, pumped protons could leak back across the membrane, disrupting the process. This switching mechanism likely involves conformational changes, so structural adjustments in the protein, that are linked to the oxidation and reduction states of certain groups. These changes can also affect the pK values of groups associated with the catalytic metal centers, making proton transfer more or less favorable depending on the state. And yes, over the years, scientists have proposed detailed mechanisms for how exactly this switching occurs. The key takeaway here is that this is a finely tuned process, where conformational shifts and redox states work together to ensure efficient proton transfer, which is essential for energy conversion in the cell. This concept ties back to our overall theme of understanding how molecular machines harness energy with precision. Alright, so in this slide, we're looking at a simplified animation that shows how protons are ejected from the mitochondrial matrix during electron transport. Specifically, for every 4 electrons transferred from 2 NADH molecules to oxygen, about 20 H+ ions are pumped out. That equates to roughly 10 H+ ions for every half O2 molecule reduced, which is a key part of how the mitochondria generate a proton gradient. Now, what's important here is that this proton pumping creates an electrochemical gradient, which is essential for ATP synthesis. The animation doesn't show it, but in reality, hydroxide ions, O, also accumulate in the matrix. They come from water dissociation, where water splits into H+ and OH. These hydroxide ions are then pumped out as well, helping maintain balance. And another point that's not depicted here is the buffering effect. The mitochondria have buffers that help manage pH and stabilize the environment, ensuring that the proton gradient is maintained efficiently. So overall, this slide highlights how electron transfer is coupled with proton pumping, establishing the electrochemical gradient that powers ATP production. It's a fundamental part of cellular respiration and energy generation. Now let's take a closer look at ATP synthase, which is embedded right in the cristae of the inner mitochondrial membrane. This enzyme complex is essential for producing ATP, the energy currency of the cell. It consists of 2 main parts, the F1 catalytic subunit and the Pho complex. The F1 part is made up of 5 different polypeptides arranged in a specific stoichiometry of A3B3JDA. This structure is responsible for the actual synthesis of ATP, converting the proton motive force into usable energy. Then there's the Pho complex, which is composed of integral membrane proteins. Its primary role is to mediate proton transport across the inner mitochondrial membrane. As protons flow through this complex, they drive the rotation and activity of the F1 subunit, ultimately leading to ATP production. Connecting this to the bigger picture, ATP synthase effectively links the process of electron transport and proton gradient formation with energy generation, exemplifying the elegance of mitochondrial function. Let's dive into how F1 Pho couples ATP synthesis to H+ transport into the mitochondrial matrix. Essentially, for each ATP molecule produced, at least 3 protons need to be transported across the membrane. This isn't just a random number. It's based on comparing two key energy values. First, we look at the free energy required for ATP synthesis under cellular conditions, which tells us how much energy we need to put in. Then, we compare that to the energy available from transferring each H+ into the matrix, considering the electrochemical H+ gradient. This gradient provides the energy that drives the process. So, in summary, the transport of a minimum of 3 protons per ATP is necessary to meet the energy demands dictated by these calculations, ensuring efficient coupling of proton movement and ATP production. This concept highlights the elegant efficiency of mitochondrial energy conversion, which is a core theme in understanding cellular respiration. Now, let's explore the chemiosmotic theory of oxidative phosphorylation, a groundbreaking concept for which Peter Mitchell was awarded the Nobel Prize. Essentially, this theory explains how ATP synthesis is coupled to respiration, but it's important to note that this coupling is indirect. Instead of a direct interaction, the process relies on a hydrogen ion, or H+, electrochemical gradient. This gradient acts like a form of stored energy, created by the movement of H+ ions across the mitochondrial membrane during respiration. As these ions flow back through specific channels, they drive the production of ATP. This idea was revolutionary because it shifted our understanding of energy transfer in cells, emphasizing the importance of electrochemical gradients. It also ties neatly into the broader theme of how cells efficiently convert energy from nutrients into usable forms like ATP. Let's delve into the chemiosmotic theory in respiration. What happens here is that electrons are transferred spontaneously through complexes I, and IV. Now, this electron transfer is coupled to a process that isn't spontaneous on its own. Specifically, the ejection of H+ ions from the mitochondrial matrix. This coupling is crucial because as H+ ions are pumped out, it creates two important things. First, a membrane potential, which we denote as dy, and it's negative inside the matrix. Second, a pH gradient, meaning the pH is more alkaline inside the matrix compared to the intermembrane space. These gradients, both electrical and chemical, are what drive ATP synthesis later on. So, in essence, the electron transfer energizes the movement of protons, setting up the conditions needed for the mitochondria to produce energy efficiently. This process beautifully links the electron transport chain with ATP synthesis, highlighting the elegance of cellular respiration. Let's delve into the chemiosmotic theory and focus on the F1-FO ATP synthase. Now, what's really interesting here is that ATP synthesis itself is non-spontaneous, meaning it requires energy input. This energy comes from the movement of protons, or H+ ions, into the mitochondrial matrix. This process is coupled to the spontaneous transport of H+, driven by the gradients created during respiration. These gradients are twofold: a pH gradient and an electrical gradient, often called the membrane potential. The key point is that respiration establishes these gradients, which then act as the driving force for H+ ions to move back into the matrix. Now, as H+ ions flow back through the FO portion of ATP synthase, they essentially use up these gradients, meaning the pH and electrical differences across the membrane decrease. This flow of protons through FO is what powers the actual synthesis of ATP. It's a beautiful example of how a gradient created by respiration is harnessed to produce the energy currency of the cell, ATP. So, once ATP is produced in the mitochondrial matrix, it needs to exit the mitochondria to be used in the cell, such as by transport pumps or kinases. This is a key step because without this export, the energy currency can't reach where it's needed. Conversely, when ATP is hydrolyzed in the cytosol, it produces ADP and Pi, which then need to re-enter the mitochondria to be converted back into ATP. This exchange is facilitated by two specific carrier proteins located in the inner mitochondrial membrane. These carriers are essential for maintaining the flow of these molecules and ensuring the continuous supply of ATP. Now, regarding the outer membrane, it's considered to be relatively permeable. Large channels called VDAC channels allow adenine nucleotides like ATP and ADP, as well as pi, to pass freely between the cytosol and the intermembrane space. Overall, this transport system is crucial because it maintains the balance of energy molecules across the mitochondrial membranes, enabling efficient energy production and utilization in the cell. Let's talk about the adenine nucleotide translocase, also known as the ADP-ATP carrier. This is an antiporter, meaning it exchanges molecules in opposite directions across the inner mitochondrial membrane. Specifically, it catalyzes the exchange of ADP for ATP. Now, at the cellular pH, ATP carries 4 negative charges, while ADP has 3 negative charges. This difference in charge is crucial because the exchange of ADP3 for ATP4 is driven by the membrane potential. In fact, this process uses up 1 charge per ATP molecule transported. So, the membrane potential not only facilitates this exchange, but also provides the energy needed for it to occur efficiently. This mechanism is essential for cellular energy distribution, connecting tightly to how mitochondria generate and export ATP to meet the cell's energy demands. Now let's talk about how phosphate re-enters the mitochondrial matrix. It does so through an electroneutral symport mechanism, which means phosphate, or pi, moves into the matrix along with a proton, H+. Importantly, this process doesn't create a charge imbalance, it's electroneutral. The entry of pi is actually driven by, and uses up, the existing pH gradient across the inner mitochondrial membrane. To put it simply, for each ATP molecule produced, about 1 mol of H+ is effectively used up to bring pi into the matrix. So, when ADP enters the matrix and ATP is synthesized, roughly 1 mol of H+ is also entering alongside pi. If we assume that the F1-FO ATP synthase transports about 3 H+ ions per ATP, then in total, around 4 H+ ions enter the matrix for each ATP generated, 3 for the synthesis process itself, and 1 used for pi uptake. This highlights how the proton gradient drives ATP production and how various steps in this process are interconnected. Understanding this helps us see the efficiency of mitochondrial energy conversion and how vital the proton motive force truly is in powering our cells. Now, let's consider the questions on this slide. First, based on the assumed number of hydrogen ions, or H+, pumped out per site as we discussed earlier, and assuming that 4 H+ ions are transferred back into the mitochondrial matrix for each ATP synthesized, what would be the predicted PO ratio? In other words, how many ATP molecules would we expect to produce per 2 electrons transferred from NADH to oxygen? This is a key point because it helps us understand the efficiency of oxidative phosphorylation. The PO ratio essentially measures how effectively the electron transport chain converts the energy from electrons into ATP. Next, let's think about how this ratio might change if the electron donor is not NADH, but instead succinate. Since succinate donates electrons through a different complex, it bypasses some of the earlier complexes in the chain, which can alter the number of H+ ions pumped, and consequently, the PO ratio. Understanding these differences is crucial, because it highlights how the source of electrons influences mitochondrial efficiency and ATP production, tying back into our overall theme of bioenergetics and cellular energy management. Alright, let's break down how we synthesize roughly 2.5 inorganic phosphate bonds, or tilde-P bonds, through oxidative phosphorylation. First, during the oxidation of NADH that's produced in the pyruvate dehydrogenase complex and the Krebs cycle, we typically generate about 2.5 tilde-P bonds. This process involves pumping about 10 protons across the mitochondrial membrane, and remember, it takes about 4 protons to produce 1 ATP molecule. So, the proton gradient directly fuels ATP synthesis here. Next, we have NADH generated in the cytosol during glycolysis. When electrons are transferred via Fad to CoQ in this pathway, approximately 1.5 tilde-P bonds are formed per NADH molecule. It's important to note that this electron transfer involves Fad and the CoQ pool, which are part of the electron transport chain's complex II. Finally, during the oxidation of QH2, which is produced in the Krebs cycle, specifically through the action of succinate dehydrogenase, another roughly 1.5 tilde p bonds are synthesized. Here, electrons are transferred via Fad and iron-sulfur clusters to coenzyme Q, completing part of the electron flow in the chain. Connecting all these points, the overall theme is how different pathways and electron donors contribute to the synthesis of ATP, emphasizing the efficiency and interconnectedness of cellular respiration. The key takeaway is that multiple sources, NADH from both mitochondrial and cytosolic origins, and QH2, feed electrons into the electron transport chain, ultimately driving the production of ATP through proton pumping. On this slide, we see how an oxygen electrode can be used to measure oxygen concentration, O2, inside a closed vessel. Essentially, as mitochondria respire, they consume oxygen, and this electrode detects those changes. When electrons are transferred, for example, from NADH to oxygen, we observe the rate at which oxygen disappears. This rate gives us crucial information about mitochondrial activity. What's interesting here is that the electrode's recording shows how oxygen consumption varies when mitochondria are respiring in the presence of inorganic phosphate, or pi, and an electron donor like succinate or another substrate that generates NADH. This setup helps us understand how mitochondria generate energy. Now, a key concept highlighted here is the dependence of the respiration rate on the availability of ADP, which is the substrate for ATP synthase. This relationship is called respiratory control. Basically, it shows us how mitochondrial respiration is tightly regulated by the cell's energy demands. More ADP means faster respiration, and less ADP slows things down. This connection is fundamental to understanding how mitochondria adapt to the cell's needs and maintain energy balance. Let's take a closer look at the respiratory control ratio. This ratio is calculated by dividing the slope after ADP addition by the slope before ADP addition. Essentially, it helps us understand how efficiently the mitochondria respond to the presence of ADP in terms of oxygen consumption. A higher ratio indicates a more effective coupling between respiration and ATP synthesis. Now, moving on to the PO ratio. This is a measure of how many moles of ADP are phosphorylated per mole of oxygen consumed. To determine this, we look at the amount of ADP added and the amount of oxygen consumed during phosphorylation. This ratio is crucial because it gives us insight into the efficiency of oxidative phosphorylation, how effectively the mitochondria convert oxygen into ATP. Both of these ratios, respiratory control and PO, are fundamental for assessing mitochondrial health and function. They connect directly to our overall theme of understanding cellular energy production and efficiency. By analyzing these parameters, we can better evaluate how well mitochondria are performing under different conditions. Let's delve into the chemiosmotic explanation of respiratory control. What's key here is that electron transfer is inherently coupled to the ejection of protons, or H+, from the mitochondrial matrix. Now, whether this coupled reaction is spontaneous depends on the combined effects of pH and electrical gradients across the membrane. When electrons transfer from NADH to oxygen, the free energy change, or DG, is negative, meaning this process tends to happen spontaneously. In fact, for this particular reaction, the DG prime is about minus 218 kilojoules per mole for 2 electrons, which indicates a strong drive for electron transfer. On the other hand, the ejection of H+ from the matrix carries a positive DG, so it requires energy input. Whether this step proceeds depends on the proton gradient. Specifically, the difference in H+ concentration, or pH, between the cytosol and the matrix, and the electrical potential across the membrane. The total energy change for the coupled process, electron transfer plus H+ ejection, is essentially the sum of these two individual components. If the overall DG is negative, then the process is spontaneous. To quantify the H+ ejection component, we use this equation. DG equals RT times the natural log of the ratio of cytosolic to matrix H+ concentrations, plus the electrical potential term, which is F times the membrane potential. Simplified, it becomes 2.3 RT times the difference in pH between the matrix and cytosol, plus that electrical potential term. So the key takeaway here is that the driving force for ATP synthesis ultimately hinges on this delicate balance of chemical and electrical gradients, all tied together through these energetic considerations. It beautifully illustrates how cellular respiration is fundamentally a process of controlled energy transduction. Alright, let's break down what happens when there's no ADP present. Without ADP, H+ ions can't flow through the faux segment of the ATP synthase. This means that the pH difference across the membrane, or dpH, and the membrane potential, dy, both reach their maximum levels. Essentially, the system hits a kind of standstill because the electrochemical gradients are at their peak. As respiration and H+ pumping continue, the free energy change, or DG, for ejecting H+ ions increases and gets closer to the DG for transferring electrons. This means that the process becomes less favorable energetically. When the coupled reaction, meaning the movement of electrons and H+, becomes non-spontaneous, respiration actually halts. We call this situation a static head because the system is essentially blocked and can't proceed forward. Interestingly, even without ADP, respiration doesn't stop completely. There's usually a low level of respiration happening, which we attribute to H+ leaks across the membrane. These leaks allow some H+ to bypass the normal flow, maintaining a minimal level of activity. This interplay between gradients, energy, and the presence or absence of ADP really highlights the tight regulation of respiration and ATP synthesis, fitting into the bigger picture of how cells efficiently manage energy production. When ADP is added to the system, it triggers a series of events within the mitochondria. Specifically, H+ ions, or protons, enter the mitochondrial matrix through the faux portion of ATP synthase. This process is what allows ATP to be synthesized from ADP and inorganic phosphate. As this happens, it leads to a reduction in the proton gradient, which we refer to as dpH, as well as a decrease in the electrochemical potential, or dy. This reduction in the proton gradient actually makes it easier for protons to be ejected, which in turn lowers the Gibbs free energy change, or DG, associated with H+ ejection. Essentially, the energy landscape shifts in a way that favors the continuation of proton movement. And here's the key point. Because the coupling between electron transfer and H+ ejection becomes more favorable, the overall process becomes spontaneous. This means the electrons can flow more freely through the electron transport chain, driving the production of ATP more efficiently. As a result, respiration, the process by which mitochondria generate energy, either resumes after a pause or is stimulated to increase activity. This entire sequence highlights how tightly linked these processes are and how the addition of ADP essentially kickstarts or boosts the mitochondria's energy production. Now, let's talk about uncoupling reagents, also known as uncouplers. These are lipid-soluble weak acids, which means they can easily pass through the lipid membranes of mitochondria. An example of such an uncoupler is dinitrophenol. In this case, a hydrogen ion, or H+, can dissociate from the hydroxyl group, the OH group, of the uncoupler molecule. This dissociation is key because it allows the H+ to be transported across the membrane. Uncouplers work by dissolving into the membrane itself and then functioning as carriers for H+. Essentially, they shuttle protons across the mitochondrial membrane, bypassing the normal protein channels that are part of the electron transport chain. This process dissipates the proton gradient, which is normally used to generate ATP, and as a result, uncouplers reduce the efficiency of oxidative phosphorylation. Connecting this to the overall theme, understanding how uncouplers operate helps us grasp how mitochondrial function can be modulated or disrupted. This has important implications in both physiology and pharmacology, especially in studying metabolic regulation and potential drug targets. Uncouplers are fascinating because they essentially block oxidative phosphorylation, which is the process mitochondria use to generate ATP. They do this by dissipating the H plus electrochemical gradient across the inner mitochondrial membrane. So, instead of protons flowing through ATP synthase to produce energy, the uncouplers allow protons to leak back into the mitochondrial matrix. This leak prevents the development of the proton motive force, specifically the proton gradient or dPH, and also prevents the electrochemical potential difference, dy. As a result, even though the electron transport chain keeps working and oxygen consumption continues, ATP synthesis is uncoupled from this process. This concept is crucial because it highlights how the proton gradient drives ATP production and how interfering with it can impact energy metabolism. When an uncoupler is present, it essentially disrupts the usual link between electron transport and ATP synthesis. In this state, there is no proton gradient, so we see no delta H or delta psi, meaning the proton motive force is effectively dissipated. As a result, the free energy change, or delta, for H plus ejection drops to zero because there's no gradient to maintain or utilize. However, interestingly, the delta for electron transfer that's coupled to H plus ejection becomes maximal and spontaneous. This indicates that electrons are still flowing through the electron transport chain, but without generating a proton gradient, the usual energy conservation processes are bypassed. This leads us to an important point. Respiration can continue even in the presence of an uncoupler, regardless of whether ADP is present or not. The process keeps going, but without the coupling to ATP synthesis, highlighting how uncouplers uncouple electron flow from energy storage. So, the key takeaway here is that uncouplers allow respiration to proceed independently of ATP production by dissipating the proton gradient and shifting the energy dynamics entirely. This helps us understand how regulation of the proton gradient impacts overall cellular energy management. So let's talk about the DG for H plus flux first. When there's no H plus gradient, the free energy change for H plus movement is zero. This means that without a difference in H plus concentration across the membrane, there's no driving force for H plus to move, and thus no flux occurs. It highlights how essential a gradient is for energy transfer. Next, the hydrolysis of ATP is spontaneous. This is a fundamental point because ATP hydrolysis releases energy naturally, without needing external input. It's this spontaneity that powers many cellular processes, making ATP the universal energy currency. Finally, in the presence of an uncoupler, the ATP synthase reaction can run backward. Normally, ATP synthase uses the proton gradient to produce ATP, but when an uncoupler is present, it collapses that gradient. As a result, instead of synthesizing ATP, the enzyme can hydrolyze ATP, running in reverse. This illustrates how disrupting the proton gradient impacts energy flow and enzyme function. Overall, these points emphasize the critical role of proton gradients and the conditions that influence energy transduction in mitochondria. Let's talk about uncoupling proteins, specifically thermogenin, which is produced in brown adipose tissue, especially in newborn mammals and those that hibernate. This protein is located in the inner mitochondrial membrane and acts as a hydrogen ion carrier. What makes it fascinating is that it essentially disrupts the usual process of creating a proton gradient across the mitochondrial membrane. Normally, this gradient drives ATP synthesis, but with uncoupling proteins active, the gradient is blocked. Instead of using the energy from respiration to produce ATP, the energy is dissipated as heat. This process, known as non-shivering thermogenesis, is quite energy-intensive. It uses up respiratory energy that would otherwise be used for ATP production, but it's incredibly valuable because it helps warm the organism, especially in cold environments. So, in summary, uncoupling proteins enable these animals to generate heat efficiently without shivering, which is crucial for survival in cold conditions. It's a perfect example of how energy metabolism can be adapted for thermoregulation.